Welcome to The Explainer. Today, we're diving into what might be one of the most revolutionary fields of our time, synthetic biology. We're moving past just observing life. Now we're learning how to actually write its code. So here's what we've got on tap. We're going to start with the big idea. What is synthetic biology really? Then we'll get a look at the actual tools scientists are using. After that, we'll explore some of the mind-blowing applications already out there. And finally, we'll tackle the big, tough ethical questions that come with all this power. All right, let's get right into it. What exactly are we talking about when we say synthetic biology? Well, to really wrap our heads around it, we have to start with one single, powerful question. Just let that sink in for a minute. What if we could program living cells just like we program computers? For basically all of human history, biology was a science of observation. We'd find stuff, name it, and try to figure out how it worked. But this is a total game changer. We're going from just reading the source code of life, all those A's, T's, C's, and G's in DNA, to actually becoming the programmers. We're writing new biological apps in the language of life itself. And that single idea, that's what's driving this entire field forward. Okay, so here's the official definition. But what does it really mean in plain English? It's where biology meets engineering. We're not just looking at life anymore, we're building with it. Scientists are taking tried and true engineering ideas, things like standard parts, modular design, quality control, the same stuff you'd use to build a car or a computer, and applying them to living cells. The goal? To create biological machines that can do things nature never even thought of. This is way beyond the old school genetic engineering, which was kind of like cutting a sentence out of one book and pasting it into another. Synthetic biology, this is like learning the language and writing a whole new library of books from scratch. So, when it comes to actually doing this, scientists basically have two main playbooks. First, there's the top-down approach. This is the practical route. You take a cell that already exists, something like E. coli, and you basically strip it down. You get rid of all the genes it doesn't absolutely need, creating this clean, predictable chassis. Think of it like the basic frame of a car. You can build all sorts of different models on top of it. Then you've got the bottom-up approach, which is, wow, way more ambitious. Here, you're trying to build a new biological system from the ground up, using non-living parts. It's like building a computer starting with just sand and copper. This approach helps us tackle one of the biggest questions out there. What's the absolute bare minimum recipe for life? A real turning point for this whole field was the idea of standardization. For biology to really become an engineering discipline, you need parts that are reliable and interchangeable. Before this, it was kind of like everyone was a custom craftsman, making their own unique screws that didn't fit anyone else's screwdriver. It was super slow and inefficient. But then came biobricks, and they changed the game. Think of them as the Legos of life. They're standardized bits of DNA that do a specific job. One might be an on switch, another might make a certain protein. You can just snap them together to build new genetic circuits. And a genetic circuit works just like an electronic one. It takes an input and gives you an output. So for example, a simple circuit could be, if you detect a toxin, then you glow green. It's this kind of predictability that lets scientists build really complex biological systems and actually expect them to work. So how is all this even possible? What's in the scientist's toolkit? Well, these tools didn't just pop up out of nowhere. They're the result of decades and decades of amazing scientific breakthroughs. Just look at this timeline. It really shows how things have just exploded. Back in 1953, Watson and Crick gave us the blueprint for DNA. And for decades, we were just figuring out how to read it. Then, bam, 2010. Scientists create the first cell that's running on a completely synthetic, computer-designed genome. That was the moment we proved, yep, we can actually write a biological operating system. Then, only 2 years later, CRISPR comes along, the so-called word processor for DNA. Suddenly, we could edit genes with unbelievable precision. And now, by 2020, we're using AI to design brand new organisms, like xenobots, these tiny living robots made from frog cells. The speed at which this is all happening, it's just incredible. So all of this technology has created this really powerful cycle. You can basically break it down into 3 steps. First, you have to read. You've got to be able to read an organism's genetic code to see how it works. And thanks to new tech, the cost of doing that has just plummeted, from billions of dollars for one human genome to just a few hundred. So we can read DNA like never before. Next, you have to write. This is DNA synthesis. It's basically like a 3D printer, but for DNA. We can now chemically create custom DNA sequences from scratch. And finally, you assemble and test it all. This is the classic engineering loop. Design, build, test, and learn. You come up with a design for a genetic circuit, you print the DNA, pop it into a cell, and see what happens. And you just keep repeating that cycle. That's what's pushing the whole field forward so fast. Okay, so with this amazing toolkit, we're moving past the what-if stage. Scientists are actually using this stuff to build solutions for some of the biggest challenges we face. These aren't just lab experiments anymore. This is happening in the real world. Take biosensors, for example. We're engineering bacteria to act as tiny living detectors. I mean, just imagine, you could spray a field with these engineered microbes and they'd only glow where there's a hidden landmine leaking explosives. Or what about a diagnostic you could drink? Like a yogurt that has bacteria programmed to detect the earliest signs of disease in your gut and then signal a problem. We're talking about turning microscopic life into these incredibly powerful, cheap tools for diagnostics and for monitoring our environment. And in pharmaceuticals, it's a total game changer. Let's talk about the anti-malarial drug artemisinin. It used to come from a plant, which made the supply chain really volatile and expensive. So what did scientists do? They took the entire genetic pathway, all 12 genes, and transplanted it into regular old baker's yeast. They basically turned breweries into microfactories, pumping out this life-saving drug cheaply and reliably. And now that biomanufacturing platform is being used for all sorts of things, from insulin to vaccines and even new antibiotics to fight superbugs. And it's not just medicine. Synthetic biology is coming for your dinner plate too. I'm talking about cellular agriculture. We're using cells to brew milk or grow actual meat completely without the animal. Companies are engineering yeast to produce milk proteins and then using those to make real cheese that's totally animal-free. And others are growing real muscle and fat cells to create cultured meat. Just think about the potential here to slash the environmental impact of farming. We're talking less land, less water, fewer greenhouse gases. This could completely reshape how we think about food. And if you think that sounds like science fiction, wait till you hear this. We're talking about 3D bioprinting, using an ink made of living cells to print out tissues like skin and cartilage. The ultimate dream? Printing whole complex organs for transplants. At the same time, you've got CAR T therapy, which literally reprograms a patient's own immune cells into living drugs. Doctors take your T cells, engineer them to hunt for cancer, and then put them back in your body. These supercharged cells then go on a search and destroy mission against cancer. It's offering real hope for diseases that were once a death sentence. Believe it or not, the applications don't even stop at Earth. NASA is looking at synthetic biology as a key technology for long-term space exploration. I mean, trying to live off the land on Mars, it's pretty much impossible without this. But imagine engineering microbes that could eat the Martian atmosphere and soil and then churn out oxygen, food, rocket fuel, even plastics for 3D printing habitats. We could be talking about the very tech that allows humans to finally set up shop on other planets. But, of course, with all this incredible power comes some really big responsibilities and risks. As we get better at engineering life, we have to get a whole lot wiser about how we use these tools. Now, when we talk about risk here, it's really important to separate it into two buckets. On one hand, you have biosafety. That's all about accidents. How do you make sure your engineered organism doesn't accidentally escape the lab and wreak havoc on the environment? People are working on things like kill switches, so these organisms can't survive in the wild. But then there's biosecurity, and that's the scarier side of the coin. This is about someone deliberately using this technology to cause harm, to engineer a superbug or a bioweapon. And that's the real challenge, right? The same exact tools that can create a cure could also be used to create a weapon. And this quote from Amy Gutman just hits the nail on the head. We're walking this incredibly fine line. How do you create rules to manage all the risks without just stopping all the amazing research that could do so much good? Every major new technology has been met with fear. The trick is to be smart about it, to be careful and put safeguards in place, but not to let fear of what could happen stop us from achieving what's possible. It's a really tough balancing act between being cautious and making progress. And at the end of the day, the biggest questions here aren't really about the science. They're about us. They're philosophical. We're messing with the definition of life and our place in the world. That whole playing God thing, it's not just a cliche. It's a real conversation we need to have about our own limits and responsibilities. And what about who gets to use this stuff? Will it just create a bigger gap between the rich and the poor, where only some people can afford these incredible new technologies? These questions don't have simple answers, and it's not just up to scientists to figure them out. We all need to be part of this conversation. So here we are, we've stepped into a completely new era. For the first time in the 4 billion year history of life, one species, us, can intentionally rewrite the code of life. We can edit it, and we can even write totally new code from the ground up. The ability to design biology it's a power that's almost hard to comprehend. The tools are here, so the question isn't, can we do this anymore? The question is, what are we going to build?